In this video, we will continue our discussion about how the Calnexin plays a role towards TrkB transportation on the cell surface. In the previous video, we have discussed that Calnexin helps in the activation of this TrkB. So in this video, we will understand the role of Calnexin in the transport of TrkB to the cell surface. So first, they checked what they did is they checked the effect of this mature TrkB data expressed on cell surface in the presence and absence of calnexin. So when they check this flow through, complete flow through, and they are able to find out that amount of TrkB data activated, amount of mature TrkB data expressed on the cell surface, the signals are very weak. So when they did the streptavidin pull down approach, so that they can find out only the TrkBs, they can see that the mature TrkBs, that is around 130 kilodaltons, are more in the case of this calnexin. When in the case of wild type calnexin, however, when the calnexins are knocked out, they can see the reduced amount of this mature TrkB. So this indicates that calnexin might plays a role towards the transport of this TrkB to the cell surface, as the mature TrkBs are reduced in amount where the calnexins are absent. And along with that, they also check this amount of this EGFR, and anti-pancalcrin act as a control, and this is the anti-calnexin to confirm that it is a calnexin knockout, and this anti-map and anti-phosphorylated map case also. So in this these are also this anti-phosphorylated map and map case helps to identify whether these signaling pathways are upregulated or not. So these are seems to be upregulated at certain conditions, such as after 1 minute and 5 minutes when the samples are acquired, both in the case of this calnexin presence and calnexin absence. However, for this anti-EGFR, what they find out is that with time, this there is a decrease in this anti-EGFR concentrations, and this could be attributed to the endocytosis that occurs when this EGF interacts with this EGFR. That thereafter helps in the activation of this TrkB, and then transport of this TrkB to the cell surface. Thereafter, transport of TrkB to the cell surface, and these things are validated using this quantitative analysis of this Western blot, where we can see that at the 130 kilodaltons, the EGF amount is significantly, the TrkB amount is significantly upregulated in the case of when the calnexin is present compared to the case where calnexin is absent. Same is the statistical significant difference has been observed at t is equal to 5 minutes. However, there is phosphorylated 100 kilodaltons phosphorylated TrkB or intercellular TrkB data found in the intercellular parts of the cells are not significantly dysregulated. However, for this EGFR, they can see they are significantly dysregulated at this zero time. However, with time, there is no statistical significant dysregulations between calnexin presence and calnexin knockout. However, the overall amount goes down with time. So this could be due to the endocytosis of the EGFR that I have just discussed. But for this 0 minute, the high amount of this EGFR receptors indicates that this calnexin positive cells has more EGFR, uh, and that leads to the activation of these calnexin present cells when they are treated with the EGF, and that cause the transport of TrkB. That could cause the transport of TrkB to the cell surface. So the reasons for this high EGFR when the calnexins are present is not explicitly explained in this paper. So now we will discuss this panel C. So in this Oh, so the one thing that I need to correct before going to the penalty is that the EGFR receptors that are expressed more when the calnexins are knocked out, not when the calnexins are present. So when these calnexin are knocked out, these EGF receptors are present more. So when they interact with this EGF, then it might activates the, it might tries to transport, activates and tries to transport this TrkB into the cell surface. However, it will get accumulated on the cell surface because of the quality control defect in the quality control mechanism of the endoplasmic reticulum, and that will cause the aggregations on the aggregations in the aggregations of this TrkB on the cells. And these things they have validated using panel C, where the immunostainings of this TrkB and DAPI has been shown. So in the case of where the calnexins are present, they can see that the TrkBs are spread across the sample. However, when this calnexin is absent, they can see that lumps of these TrkBs that are formed. And when they quantify these immunostaining images, they can find out that TrkB clusters per cells are uh, statistically low compared to the case where these calnexins or knockouts are present. So clusters of TrkBs found to be more, and this could be because of the high EGFR that are present in the calnexin knockout cells. And when they check the cluster size, that is also statistically significant, that in the case of calnexin knockout, they can see the high cluster size of the TrkB compared to the case of where these TrkBs are compared to the case where this calnexin has been not been knocked out. In order to further validate this, they stain the cells with this M-cherry KDL sequence that can identify or that can stain the endoplasmic reticulum. And for when the calnexins are present, for this they can see that the TrkBs are spread across the endoplasmic reticulum. However, when these calnexins are absent, they can see that there is a lumps formations of this TrkB occurred within the cells or within this endoplasmic reticulums also, that can be seen here. However, for when the calnexins are present, there is no lump formations that has been observed. And when they quantify biochemical analysis of this images shows that this TrkB and M-cherry cattles are overlap when these calnexins are present. However, they are not overlapping, indicates that there is lump formations occurs. There is a kind of lump formation occurs when the calnexins are absent, and this they have attributed this to the defect that has occurred in the quality control of this endoplasmic reticulum because of the absence of calnexin. Calnexin cal-reticulum cycle plays important roles towards providing the native structures of the glycoproteins. So I hope you now understood why how they validated their hypothesis of calnexin coordinates the transport of TrkB on the cell surface from the endoplasmic reticulum. And if you need to understand more about this paper in detail or this experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.